Hello everybody, welcome to Deeksha Online. Now in today's class, we will be starting a new chapter. The name of the chapter is Sexual Reproduction in Flowering Plants. So let us go through a quick roadmap or the chapter overview. Now in this particular chapter, you will be discussing about the flower. Now you all know that the flower is the reproductive organ in case of flowering plants. The flowering plants are nothing but the angiosperms. We shall see what are the different structures and what are the events that happen in the flower before the process of fertilization, that is pre-fertilization event. And then we shall see the process of the fusion between the gamete and the egg, that is referred to as syngamy, which is a part of a much complex process that is referred to as double fertilization. Then we shall see about what happens after fertilization in the flower, what are all the changes that happen in the plant body, and what are the structures and events associated with the same in the post-fertilization events. And lastly, we will be discussing about certain aspects such as apomixis and polyembryony. So this gives you an idea of what are all the things you can look forward to in this particular chapter. Now you all have learnt in your 11th standard what is a flower. So a flower, as you all know, is born only in angiosperm plants, which are the highly advanced vascular plants, and these flowers are nothing but the reproductive structures. Let us do a quick recap of what are the different parts of the flower. So in the flower you have the non-essential or the accessory parts, and you have the essential or the reproductive parts which actually take part in reproduction, which actually take part in the formation of seed, isn't it? So we can classify the parts of the flower into non-essential part and essential part. The two major non-essential parts are sepals and petals, and the two major essential or reproductive parts are androecium and the gynoecium. The androecium is considered to be the male reproductive part of the flower, and please remember the gynoecium is considered to be the female reproductive part of the flower. And what are the units of androecium? The units of androecium are referred to as the stamen, and the units of gynoecium are referred to as carpel or pistil. So in this chapter, we will be focusing mainly on the essential parts or the reproductive parts of the flower, which include the male reproductive part, which is called the androecium, and the female reproductive part, which is referred to as the gynoecium. So this is a diagrammatic representation of a longitudinal section of the flower. Now here you can see the sepal, that is the calyx. The entire ring of sepals is referred to as the calyx, and then you can see the petal. The entire ring or whorl of petals is referred to as the corolla. So the sepal and the petal make up the units of the accessory or the non-essential whorls of the flower or the non-essential parts of the flower. And now you can see the structure that is sitting in the center of the flower, and this structure is referred to as the gynoecium. The part of the gynoecium that is visible to you, the unit of the gynoecium that is visible to you here, is referred to as the carpel. So you all are aware that a carpel has an upper expanded disc-like structure and a narrow neck-like structure and a basal part which is swollen like this, isn't it? And it is present on the base of the flower, as in the thalamus of the flower. And all of you are aware of one more thing, that so see the labelings over here. This upper expanded part of the carpel is referred to as the stigma, and then the narrow neck is referred to as the style, and the lower swollen part is referred to as the ovary. So what do we call this entire structure which I have shown here? This entire structure is the unit of, it is a unit of gynoecium. Remember that gynoecium is the female reproductive part of the flower, and this unit of gynoecium is referred to as a carpel, or it is referred to as a pistil. Is that clear to all of you? So a carpel or the pistil has three important parts. The first part is this stigma, and then the second part is the neck, which is referred to as the style, and the third part is the swollen part, which is the basal swollen part, which is referred to as the ovary. Is that clear? Now, inside the ovary, we had discussed that there is a structure, a spore producing structure that is sitting inside the ovary, and this structure, which plays a very important role in the process of reproduction, is referred to as the ovule. The technical term for the ovule is the megasporangium. So sitting inside the ovary is nothing but the ovule. If you remember from the chapter of reproduction in organisms, we had seen that the ovary wall becomes the fruit wall. The wall of the fruit is referred to as the pericarp, and the ovule after fertilization becomes the seed. Okay, so I hope you remember that the wall of the ovary becomes the fruit wall or the pericarp, and the ovule itself transforms into a seed that is after the process of fertilization. So this part of the flower is referred to as the carpel or the pistil. It is a part what is referred to as technically the gynoecium, which is the female reproductive part of the flower. Now let us see the male reproductive part of the flower. Now the male reproductive part of the flower consists of a structure which has an elongated filament, and at the end of the filament there is a bilobed structure like this, and this bilobed structure is referred to as the anther, and this thread-like structure is referred to as the filament. Now, what do you call the anther and the filament together? The anther and the filament together is referred to as the stamen, and the stamen is nothing but the unit of the male reproductive flower, the unit of what is referred to as the male reproductive part of the flower, which is called the androecium. Clear? So this is the unit of androecium, and what do we call this unit of androecium? We refer to it as the stamen. So I hope it is clear. We have now currently discussed the two major essential walls of the flower. The first essential wall of the flower I have discussed with you is the gynoecium, which is made up of units called pistil or carpel, and then the second one that we have discussed here is nothing but the androecium. The unit of androecium is referred to as the stamen. The upper part of the stamen has the bilobed anther, and the thread-like structure, the filamentous structure, is simply referred to as the filament. Okay, so this is the two major. So this was first one and this was the second one, the two major essential walls, the essential parts of the flower. Why are we calling them essential? If you recall, we call them essential because they are the parts of the flower which actually participate in reproduction, and therefore they are called the reproductive structures of the flower or the essential walls of the flower. Okay. Now, when a plant is ready to reproduce after it has completed its vegetative growth, there are several hormonal changes that happen in the body of the plant, and several structural changes that happen, and ultimately there is differentiation at the tip of the stem. Mostly the shoot apical meristem modifies itself into a structure that is referred to as the floral primordium. The floral primordium is the tissue which ultimately takes the shape of or gives rise to the flower. So you all know that a plant does not flower from the beginning, isn't it? It goes through a period where it increases in its size, it grows, it attains a certain degree of maturity, and that also we had learnt in the chapter Reproduction in Organisms, that this period in the life of a plant is called the vegetative period, or the vegetative phase or the vegetative period. After that, there are several changes happening in the plant body, where the meristem present at the tip of the plant body, the tip of the stem, will transform into a floral primordium, which will ultimately give rise to a flower. So if this is the shoot tip, this is just for your understanding. These are the branches of the plant. Okay, now if this is the shoot tip, there is a tissue here which is referred to as, or you can say that there are a lot of actively dividing cells over here at the tip of the plant, which will ultimately take the shape of a flower, and then ultimately it becomes a floral primordium, and this floral primordium will ultimately become what flower that you saw in the previous diagram, with petals and with sepals and with ovary and the female part and then we had seen the male part, which is referred to as the androecium. So this is how the floral primordium will take the shape of a flower when the plant has become reproductively or sexually mature. Usually the flower may be borne singly, like how I have shown in the diagram. This is the tip or the shoot apical meristem, or the meristem present at the end of a branch or the end of the plant itself. It may transform into the floral primordium, which may transform into one single flower, then we refer to it as a solitary, or it may transform into an axis which may have many flowers attached to it, isn't it? So when it has many flowers attached to it, we call these smaller flowers as florets, and we call such an aggregation of florets, we refer to it as inflorescence. So flower is either born singly, as in this particular case, or it is born as a group. When flowers are born as a group, they are referred to as the inflorescence. Is it clear? So flower may be solitary or it may be an inflorescence, and it develops from a special type of tissue that is referred to as the floral primordium. We have already discussed that the flower has a male reproductive structure. The male reproductive structure of the flower is called the androecium. The female reproductive structure of the flower is referred to as the gynoecium. I have already told you that the units of androecium are referred to as the stamen, and the units of gynoecium are referred to as the pistil or carpel. Okay, we have also seen the parts of the stamen. The stamen has two parts, that is anther and filament. The pistil or the carpel has three parts, that is stigma, the style, as well as the swollen part, which is referred to as the ovary. Okay, now we shall be focusing our attention mostly on the male reproductive part, that is the unit of androecium, that is referred to as the stamen. So what are the two parts of the stamen? We saw that there is an elongated stalk or a filament, and then at the end of the filament there is a bilobed structure that is referred to as the anther. And where is the filament attached? The filament is attached to the structure on which all the parts of the flower are situated, that is referred to as the thalamus, or a petal of a flower on one side and bears a bilobed structure which is referred to as the anther. So please remember that the filament is usually attached to the thalamus. Now if you say that this is the flower, the expanded part of the flower that is above the stalk is referred to as the thalamus, and it is to this that the filament of the stamen is attached. So this is the long stalk, which is the filament, and these bilobed structures are nothing but the anthers. And what is this expanded part at the base of the flower on which all the parts are situated? This is referred to as the thalamus or the receptacle. Alright, and this is the stalk of the flower. And what do you call the stalk of the flower? The stalk of the flower is referred to as the pedicel. So this is the attachment of the stamen, and now see at the upper part of the stamen, like I told you, there is a bilobed structure which is referred to as the anther, and then you can see the filament over here. Now why is anther considered to be a bilobed structure? Now all of you look at this diagram here, you can see that they have taken a cross section of the anther. It's like they have cut through the anther like this, and this is what is visible when you take a cross section of the anther. Now, when you take a cross section of the anther, you can notice you can draw an imaginary line passing through the center of the anther, like what I'm showing you here, and you can see there is one half of the anther this side and one half of the anther the other side. So on the either sides of this imaginary line, since there are two parts, we can call these halves as the lobes of the anther. Since the anther has two lobes, we say that the anther is bilobed. Did you understand? And this imaginary line which I have shown over here, if you draw an imaginary line, you can see that there are two lobes, and that's why we say that the anther is considered to be a bilobed structure. So say for example, let me draw a simplified diagram over here. Now, so this is one half, this is the second half of the anther. Okay, so imagine you have taken a section of the anther. So you can see this is the part of the anther which I am showing you here, the sectional view of the anther. This is the complete anther which is being shown over here. So what are the two halves? I told you that you have, if you consider an imaginary line passing through the center. And on what basis did I consider this imaginary line? Because in the center of the anther there will be a special tissue which I am shading here, which I am drawing striations here. This tissue is referred to as the connective, because it connects the two halves of the anther. Okay, so here you have taken a transverse section. In the transverse section, in the center of the anther, you can see a tissue, a sterile tissue, which is referred to as the connective. Now on either sides of the connective, this will be one lobe of the anther and this will be the second lobe of the anther. So 1 and 2. So because there are two lobes, because there are two halves on the either sides of the connective, we say that the anther is considered to be a bilobed structure. Is that clear to all of you? So what are the two parts of the stamen? Let us write it here for convenience. Please write down in your books. You have anther. Anther has how many lobes? It has two lobes. On what basis are you calling it lobes? Because you are considering the connective tissue as a reference. Connective is a sterile tissue present in the center. On both sides of the connective, you have two halves which are referred to as the two lobes, and that's why we call the anther as a bilobed structure. And then what is the stalk of the anther called? The stalk of the stamen, the stalk of the stamen is referred to as the filament. At the end of the filament is where you can see that the anther is located. Okay. Now again we are going to focus on the section of the anther, because we need to learn the structure of the anther. For you to understand the structure of the anther, you obviously will have to keep taking transverse sections. I told you that there are two halves on the either sides of the connective tissue, and that is why anthers are called bilobed. Now why do we call anther as a dithecous structure? Now once again you can see the section here, or I will just draw it once again for you. So remember, so you have taken a transverse section of the anther. Okay, so this is the transverse section of the anther. You have cut the anther into half and you are seeing what is there inside the half of the anther. Now I told you the sterile tissue that is present in the center is referred to as the connective. Now this half of the connective, on the one side of the connective is one lobe, and this part of the anther on the other side of the connective is the second lobe. So how many lobes are there in the anther? Two lobes are there. Now let us see why we call it as dithecous. Focus on one lobe. In each lobe you will see that there are two compartments or two cases. Please remember I am using the word cases. So there are two cases, 1 and 2 in this half of the anther, and then there are two cases again, 1 and 2 in this half of the anther. Correct? So totally how many cases are there? 4. But in each lobe of the anther, how many cases or how many compartments are there? 2. That's why we say that there are two thecae. Now theca is a Greek word. It is derived from the Greek word that is thecae, which means a case or a compartment. Alright, so in each lobe you are seeing how many compartments now? You are seeing two compartments. So I'm going to label these compartments or these chambers as the thecae. Later you will see what this chamber exactly is all about. For now, because I'm trying to explain what a dithecous anther means, you have to remember that in each half, in each lobe of the anther, you have how many thecae? You have two theca, and that's why we call this anther as a dithecous structure. Again, to see the thecae, what should you do? You should take, like I have taken here, you have to take the transverse section of the anther. In the diagram of the textbook also, let me roughly highlight the thecae. See, this is one theca here. This is the second theca in this lobe of the anther. This is the one theca. This is the second theca in this lobe of the anther. So this is one lobe. This is the second lobe. So one half and the second half, that's why we call the anther as bilobed. Each half has two compartments or two cases or two thecae. That's why we refer to the anther as a dithecous structure. Is that clear to all of you? Okay. Now moving on to the structure of the anther again. I told you that there is a bilobed nature. It becomes very, very distinct obviously when you take a transverse section of the anther. And the anther, when you take a transverse section, it appears to be four-sided. Let us highlight the four sides. This is side 1. This is side 2. This is side 3. And this is side 4. So totally how many sides are there to the anther when you take a section? Four-sided. That's why we say that anther has a tetragonal structure. And how many cases or thecae are there? There are 4. 2 in each half. 2 in each lobe. That's why if you remember we called it as dithecous. But in totality how many cases or compartments are there? 4. And what is the name given to this case or compartment? The name given to this case or compartment is the microsporangium. Okay, so totally there are 2 in each lobe. Totally throughout the anther you have 4 cases or 4 compartments which are referred to as microsporangia. Once again I am going to draw the section of the anther so that you don't get confused. All of you see here, again we have taken a section of the anther. Now this is the part of the anther which is not sectioned, so obviously to show you continuity, this is the filament. Okay, so this is the section of the anther. Now what is this tissue I am drawing? I am drawing this again and again so that you are comfortable with the concept. So this is the connective, so I am going to label this sterile tissue in between as the connective. Then this is what? This is lobe 1. This half of the anther is lobe 1 on this side of the connective, and on the other side of the connective there is what? There is lobe 2. Now because there are 2 lobes, what do we call this anther as? We call it as a bilobed anther. Yes or no? Now let us look into each lobe. Inside each lobe, how many compartments are there? 1, 2. So in this lobe, how many compartments are there? 1, 2. So totally how many compartments? 1, 2, 3, 4. And what are these compartments called? Literally they are called theca, because in Greek the word theca means a case or a compartment. But now we know that each structure, each chamber or each compartment or each case that is located inside the lobes of the anther is referred to as a microsporangium, and here it is very clearly mentioned there are 2 microsporangium in each lobe of the anther. Okay, so please remember this microsporangium. Why is it looking circular? Because you have taken a section. In reality, the microsporangium is a cylinder which extends throughout the length of the anther. See, it is a cylindrical compartment. But why is it looking circular here? Because you have taken a section. If you cut a cylinder, it will look circular in outline. So this each of these tubes, each of this thecae or microsporangia is a continuous tube-like structure, a cylindrical structure which extends throughout the length of the anther. See how it is extending throughout the anther here. So it is actually a cylinder. Because you have taken a section, they are looking what? They are looking circular in outline. So please remember, they are either called a case or a compartment, in which case we should refer to it as a theca. But technically what are these thecae referred to as? They are special structures called microsporangium. Let us see what is there inside each microsporangium. Totally how many microsporangium are there? 4. That's why we call a typical dithecous anther as what? We call it as, very, very important, we call it as tetrasporangiate. Tetra means four. Four microsporangia are present in the anther. That's why we call it as tetrasporangiate. And like I told you, each microsporangium is not just a circular chamber, it is an elongated cylindrical chamber which extends throughout the anther. It looks circular because you have taken a cross section. Is that clear? What do the microsporangia become later? When the anther matures, now this diagram is apt for this. When the anther matures, the microsporangia will become something called the pollen sacs because they are filled with the pollen grain. Okay, see how the pollen grain are it is overflowing with pollen grain, isn't it? So this is what happens to each of the microsporangium once it becomes mature, once the anther becomes mature. So how many microsporangia are there in a dithecous or a bilobed anther? There are totally 4 microsporangium, and ultimately each of these microsporangium will become what? They will get filled with pollen grains, and we should now in the later stages call them as what? We should in the later stages we should call them as pollen sacs. That means in each half of the anther, I will be drawing this time and again. Okay, so you have taken a section, so then remember this is the section of the anther. There's 1, there's 2, there's 3, there's 4, 2 each lobe, and then now you can see they're filled with pollen grains, and so now you will call these microsporangia which are filled with pollen grains in them, we refer to them as the pollen sacs. And like I told you, they are cylindrical, they extend throughout the length of the anther. They are not just a circular structure, they're actually cylindrical structures. See how it is extending throughout the length of the anther, correct? Each cylindrical structure is a theca or a microsporangium, and this microsporangium later, what does it become? It is filled with pollen grains. So when the anther is mature, we should now call the microsporangium as a pollen sac, and you know the connective that is present in between, which joins the lobe on the left side and the lobe on the right side. Okay, now we shall look into the TS of a young anther, what we've been doing all along, but we will look into the structure of microsporangium. How exactly does the microsporangium look like? Again, for that you have to see, now you can see these 4 microsporangia. There's 1 here, there's 2 over here, there's 3 over here, isn't it? And this is the 4th one. So totally how many compartments are there? How many cases are there in the anther? There are 4 microsporangia. That's why I told you it is called what? It is called a tetrasporangiate anther. Okay, so when you take a transverse section, I had told you each microsporangium or the compartment appears to be circular in outline because it is a cylindrical tube. If you take a section, it will look circular. And remember, in a young anther, it is surrounded by 4 major layers, starting from the outer epidermis to the inner endothecium. Inner to that is the middle layer, and the innermost cell layer of the microsporangium is referred to as the tapetum. See what they have done here. Now see this diagram. This is the section of the young anther, and they have highlighted the wall of a portion of the wall along with the microsporangium for you over here. Now see the layers in the wall of the anther. The outermost layer is the epidermis. This is the epidermis, correct? Inner to the epidermis you have the endothecium, and then you have middle cell layers, the middle layers, and this is the innermost layer of the that is the actual microsporangium that you can see, which is referred to as the tapetum. So how many layers are there? That is, the microsporangium is surrounded by how many layers? There are 4 layers in the wall of the anther. The outer epidermis, inner to that is the endothecium, inner to that is the middle layer, and inner to that is the tapetum. It is very important to note that some of the cells of the tapetum will have 2 nuclei in them, and this layer which has cells bearing 2 nuclei with very dense cytoplasm is referred to as the tapetum. See, so this is how a portion of the anther will look like, a young anther will look like if you magnify a part of the microsporangium and the layers of cells that surround the microsporangium. Did you understand what I'm saying? So how many cell layers are surrounding this each microsporangium? Remember, there are 4 microsporangia, that is 1, 2, 3 and 4. Each microsporangium is surrounded by how many layers? 4 layers. What are those 4 layers? Outermost epidermis, then inner to that is endothecium, inner to that is middle layers, which may be more than one, and the innermost layer is referred to as the tapetum. And now can you all see, I'm going to highlight a group of cells which are sitting inside the microsporangium. See these group of cells. I'm going to draw an outline to the boundary of this group of cells. This entire group of cells which have nucleus, a single nucleus in them, and which is sitting inside the microsporangium, is nothing but a mass of cells. And can you see that all of these cells are alike? Now here you ignore this labeling for time being, I'll come to this later, but this entire tissue which is made up of cells which look very much alike, which are present in the microsporangium, these are referred to as the sporogenous tissue. Okay, so what is this mass of alike looking cells called? This mass of like looking cells are referred to as the sporogenous tissue. Why have they labeled a cell of this sporogenous tissue as a microspore mother cell? That we will come to in the next slide. So for now, in this slide, what did you understand? In this slide, you took a section of the anther, you saw the microsporangium. The microsporangium has a central mass of homogeneous cells which are alike, and this tissue is called the sporogenous tissue. And in the microsporangium, the sporogenous tissue is surrounded by how many layers? Is surrounded by 4 layers. What are the 4 layers? Outer epidermis, inner endothecium, middle layers, and the innermost layer, that is referred to as the tapetum. Okay. So what is the function of the outer 3 layers? That is the epidermis, the endothecium and the middle layers. They are mainly meant to protect the tissue of the microsporangium, and they also cause the breakdown of the anther. The anther will rupture by a process called dehiscence, so that once the pollen grain are formed, they need to be liberated to the outside. So these layers, the outer 3 layers of the anther surrounding the microsporangium, surrounding the sporogenous tissue which makes up the microsporangium, is nothing but meant for protection. What about the innermost wall layer of the microsporangium, that is surrounding the microsporangium? We saw that the sporogenous tissue is immediately surrounded by a layer called tapetum, which I told you has binucleate cells with lot of dense cytoplasm. They are the ones which help in the nourishment of the developing pollen grains. Pollen grains are produced inside the microsporangium, which derive their nourishment from this layer which is referred to as the tapetum. The tapetal cells, like I told you, they have a dense cytoplasm and they have more than one nucleus in them. So once again, you can all see when the anther is young. Okay, so I told you when the anther is young, there is a group of homogeneous tissue which occupies the center of the microsporangium, surrounded by these 4 layers which we talked of. So remember, when the anther is young, inside this microsporangium, there is a layer, there is a group of cells which all look alike. And what is this tissue which is a mass of cells which look alike called? Now this is what is referred to as the sporogenous tissue I had labeled in the previous slide. So when the microsporangium is still in a young anther, it consists of, what does it consist of? It consists of a mass of cells which are alike to each other. See, all the cells look alike, they have a single nucleus, and this is referred to as the sporogenous tissue. It occupies the center of each microsporangium. Each microsporangium means each case or theca. By now you know a microsporangium has two things. It has a wall and it has a group of cells inside. Now by now you know that the wall of the microsporangium has how many layers? It has 4 layers. The innermost wall is the tapetum, which provides nutrition to the developing pollen grains, and in the center of the microsporangium you have a mass of like cells which look similar, which is homogeneous, and this is referred to as the sporogenous tissue. Okay. What is this sporogenous tissue? What is the speciality of this sporogenous tissue? So remember, these four are the wall layers, and the central mass of tissue that is sitting in the center of the microsporangium is now referred to as the sporogenous tissue. Here they have labeled one particular cell of the sporogenous tissue. So each cell of the sporogenous tissue can be referred to as a microspore mother cell, or it can be referred to as a pollen mother cell. Pollen mother cell is abbreviated as PMC, and these cells are diploid. They have they have two sets of chromosomes in them. So remember, every single cell in the sporogenous tissue, not only this which I have highlighted, this as well, this as well, this as well, this as well, this, every single cell within the sporogenous tissue is a potential microspore mother cell or a pollen mother cell. Now, why is it called a microspore mother cell? Because this cell which is present in the sporogenous tissue has the ability to take part in meiosis. So it is diploid, but this cell, which is the microspore, I'm highlighting here also, this microspore mother cell, which is sitting inside as the sporogenous tissue, is capable of undergoing meiosis. You all know when a cell undergoes meiosis, we get 4 haploid daughter cells. So this daughter cell cluster that is formed, which is when this cell undergoes the meiosis, surprisingly all the daughter cells are aggregated in the form of a cluster of 4. See, 4 daughter cells which are sitting together in a group of 4. We can call this cluster as a tetrad. And what is each daughter cell referred to as? Each daughter cell is referred to as a microspore. And this microspore has now become haploid. Why has it become haploid? Because it is a product of meiosis. And who underwent meiosis to form this cluster of 4 haploid microspores? It is each cell of the sporogenous tissue. In this, I have taken only one cell. So this cell of the sporogenous tissue, which is referred to as the microspore mother cell, undergoes meiosis to form 4 haploid daughter cells called microspores, which are stuck together to form an aggregate called, so let me call this as a microspore tetrad. Now you know what will happen to this microspore tetrad as the anther becomes mature. As the anther becomes mature and as it starts dehydrating, mature plus as the anther grows, what will happen is it loses much of the fluid. Okay, so in mature and dehydrating anther, what will happen is all these 4 cells will split from each other. See, now I'm going to show these 4 cells as if they have separated from each other. They are no longer stuck to each other. So what have they become now? They have become free microspores. Here they were a microspore tetrad, but once the anther starts maturing, they start getting separated from one another. And what will happen to each one of them? Each one of them will mature into a pollen grain. Did you understand? So each of these microspores will further undergo a maturation, that is, will divide by mitosis. We'll come to that later, and they will give rise to a pollen grain. So how many pollen grains am I getting here? 1, 2, 3, 4. There are 4 microspores, so obviously I will get 4 pollen grains from each. And when do these microspores separate from each other, and when do they give rise to pollen grain? When the anther becomes mature and when it starts undergoing dehydration. Okay, I hope this is clear to all of you. So once again, what are the parts of the microsporangium? They have the wall layers, and they have a central tissue which occupies the center of the microsporangium. The center of the microsporangium is referred to as the sporogenous tissue. In the sporogenous tissue, each cell is a potential what? A microspore mother cell or a pollen mother cell. Is it haploid or diploid? It is diploid. It undergoes what? It undergoes meiosis to form a group of how many haploid daughter cells? To form a group of 4 haploid daughter cells. What are the daughter cells called? They are called microspores. How are they aggregated? In the form of a microspore tetrad. Later the tetrads get separated from one another, and what happens to each microspore that came from the tetrad? Each microspore will mature into a pollen grain. So from one tetrad, how many pollen grains are originating? 4 pollen grains originate from one tetrad. Is this clear to all of you? This process where what did we get? We got, or we started off with, a microspore. Please see this carefully. We started off with microspore mother cell which was diploid. What did it undergo? It underwent meiosis to form haploid what? To form haploid microspore, single or tetrad, to form a tetrad of haploid microspore. This phenomenon is called microsporogenesis. The process where the microspore mother cell or pollen mother cell through meiosis gives rise to haploid microspores. Please remember the microspores are haploid, and this phenomenon is referred to as the microsporogenesis. Where is this microsporogenesis happening? It is happening inside the microsporangium. What do you call the cells of the microsporangium? Sporogenous tissue. Each cell is a potential what? Each cell is a potential microspore mother cell, or it is a potential pollen mother cell. What happens to all the microspores when they get separated? They start maturing into the pollen grain. So one microspore mother cell is producing 4 haploid microspores. Each one is developing into pollen grain. So we get how many pollen grains from one haploid? From one microspore mother cell or pollen mother cell, we get 4 haploid pollen from 4 haploid microspore tetrad. Did you understand what I'm saying here? Okay. This process of formation, what did we get in the end? Haploid microspores. From whom? From diploid pollen mother cell or microspore mother cell, who is a cell of what tissue in the center of the microsporangium? The sporogenous tissue. The peripheral region is the wall layers, I told you. The central region of the microsporangium occupies what? It occupies the microsporangial tissue, which is called the sporogenous tissue. Isn't it? So formation of haploid microspore from the diploid pollen mother cell, through which process? Through meiosis, is referred to as microsporogenesis. See, I told you that the microspores that are formed are arranged in a cluster of 4. What are they called? A microspore tetrad. As the anther matures and dehydrates, the microspores separate. Each microspore matures into what? They mature into a pollen grain. So once again, I'm going to show you here. This is the microspore mother cell. I told you that the microspore mother cell is capable of what? Is capable of meiosis. When it undergoes meiosis, it forms a cluster of 4 cells, which is called a microspore tetrad. Then as the anther matures and dehydrates, these 4 cells will become separated from one another. See? They are getting separated from one another. They are becoming independent, and each one develops into a pollen grain. Isn't it? So totally we have one microspore mother cell is capable of giving rise to 4 microspores, and 4 microspore, each of the 4 will transform into 4 pollen grains. So therefore one microspore mother cell ultimately gives rise to a set of 4 mature pollen grains. But remember, this stage where all the microspores are adhered into a cluster is referred to as the microspore tetrad. Okay? So from this it is clear that the job of the anther is to produce pollen grain. Inside each microsporangium, thousands of pollen grain are produced, and now see, this is the mature anther. Once the anther matures, how the anther is rupturing. When the anther breaks open, we say that this phenomenon is referred to as the dehiscence. So now when the anther dehisces, you can see how all the pollen grain will be released out to the outside environment. So you can see how the pollen grains are formed inside. Now you know how the pollen grain came from. They came from the pollen mother cell or the microspore mother cell within the TK. What is that TK or compartment called? Microsporangium. Microsporangium has two layers, a sporangial tissue in the center, a sporogenous tissue, and a wall. The wall has how many layers? The wall has 4 layers. So if they ask you which is the innermost layer in the wall of a young anther, you have to say it is tapetum, which nourishes the sporogenous tissue and the pollen grain that are formed inside the anther. Okay. So I told you that they form a cluster of 4. So what does this diagram indicate? This diagram is nothing but a microspore tetrad. And then I told you very soon the 4 cells will get separated from each other. See, they are no longer attached to each other. And what are they now called? They are simply called a microspore. So this is how a microspore looks like. So it becomes spherical. So I'm going to label this as the microspore, which is haploid. Where initially it was what? It was a part of the tetrad. Now it has become a single microspore because it split from its partners. It was found in a group of 4. Now it splits from its group, and therefore now this particular microspore has now become alone. So it is a single microspore. It is no longer a part of the microspore tetrad. Now see what is happening to the single microspore. Now this microspore will develop vacuoles in it. So this is the continuation of the diagram. After this you follow the arrows which I am showing over here. This is the continuation of the diagram. They develop vacuoles in them. Notice how the nucleus of the microspore is getting pushed towards one side of the cell. It is not in the center of the cell. So it moves towards one side and then it undergoes mitosis. The nucleus undergoes mitosis. See, this is the spindle fiber. Did you notice that the spindle fiber, see, normally in a cell, if you see, the spindle fiber is occupying a central position. These are the spindles and the chromosomes are aligning at the center. It occupies a central position. But in this cell, if you notice, the spindle is formed somewhere at one pole away from the center. See, here it is central in position, but in this case the spindle is being pushed from the center. So when we say this kind of a cell division where the spindle is not formed in the center of the cell, we call it as the asymmetric spindle. Because of this mitosis, now did you notice, so the microspore is undergoing mitosis. Because of this, 2 cells are formed inside. There is a larger cell. This larger cell is referred to as the vegetative cell. There is a tiny cell that is sitting here with very little cytoplasm in it, which is now called the generative cell. So what are the 2 cells that are formed by? Why is one cell larger? So this is the larger cell and the generative cell is smaller. So can you think and tell me why one cell is larger and one cell is smaller? That is because of the asymmetric spindle. The spindle was formed on one side in an asymmetric position. So one of the cell becomes larger in size and one of the cell becomes very smaller in size. Now this is what we refer to as a pollen. So a pollen grain usually in 60% of the cases it usually has 2 cells in it. What are the 2 cells of the pollen grain? A larger cell which is referred to as the vegetative cell, a smaller cell which is referred to as the generative cell. Did you understand the mechanism? So what did we start off with? We started off with microspore tetrad. Then I told you that the tetrads get separated into singles. Then each single cell is now again called as a microspore. What happens to the microspore? It develops vacuoles. Its nucleus moves towards one end, and that nucleus undergoes mitosis through what type of a spindle? Asymmetric spindle away from the center, and it produces one larger cell and one smaller cell, which is called vegetative and generative respectively. And this structure, which has these two cells in it, is now referred to as a pollen grain. So you saw how a microspore, which had only one nucleus in it, has transformed itself into a pollen grain which has two cells in it, a larger cell which is called the vegetative cell, and a smaller cell which is referred to as the generative cell. So this is a pollen. This two-celled structure is the pollen, and this is the microspore. So now we saw how the microspore develops into a two-celled structure, which is a two-celled again a cell which has another cell inside, so it becomes a two-celled structure which is referred to as the pollen grain. And then the diameter of the pollen grain is about 25 to 50 micron meter in diameter. So remember, a microspore has given rise to a pollen grain which is about 25 to 50 micron meter in diameter. Clear now? This is not just it. We have to learn the details of the pollen structure. In this diagram as well as in this diagram, I have not given the details of the structure of the pollen grain. So now you saw how each microspore develops into a two-celled structure which is referred to as the pollen grain. See the actual structure here. Again, I have taken the textbook diagram only. I will be making some modifications to this. So actually in a pollen grain, you are seeing two cells. The larger cell is called the vegetative cell. The smaller cell is somewhat flattened and bulged in the center, and it occupies one position in the vegetative cell. You know that there is one large and one small. So the vegetative cell is larger and the generative cell is smaller. That is because of the asymmetric spindle. Now, on the outside of the pollen grain, a very thick and resistant wall will develop, like what I'm showing you here. I'm showing this wall in a specific pattern like this because it is a wall which has lot of patterns and designs on it. It is very wonderfully ornamented. Okay, it is not just a smooth surface, and this region in the pollen grain, the outermost wall of the pollen grain, is referred to as the exine. So remember, the outermost wall, which is thrown into ridges, which is thrown into projections, which is very wonderfully ornamented. It has all sorts of patterns on it, and this wall, which I have shown in red over here, which is the outermost protective covering of the pollen grain, is referred to as the exine. Now can you see that there is a wall inside, which I am going to highlight in red now. The inner wall, which is a continuous layer, it is also a cell wall. It is a continuous layer made up of cellulose and pectin, and this inner layer, which is made up of cellulose and pectin, which is a continuous layer, is called as the intine. Why are we calling this as the continuous layer? Does it mean that the exine is discontinuous? Yes. Can you see these areas where exine is missing? See, exine is absent over here. Here also exine is absent. Only intine is there, but exine is not there. Such areas are referred to as the germ pores. Did you understand? So regions of the cell wall of a pollen grain where what is present and what is absent. In these regions, exine is absent. Only intine is there. And such areas in the pollen grain where exine is absent and intine is present, it is referred to as the germ furrow or the germ pore. And then you can see an inner layer, which I am going to show in dotted line. This inner layer is nothing but the cell membrane of the vegetative. The largest cell which I am highlighting now is nothing but the vegetative cell. So this inner layer is referred to as the plasma membrane. Now let us see inside the pollen grain. So you have a cell. This cell is referred to as the vegetative cell, and this tiny cell sitting below is referred to as the generative cell. Notice how the vegetative cell has an irregular shaped nucleus. The nucleus is irregular shaped. It has lot of dense cytoplasm in it, isn't it? So the vegetative cell has irregular nucleus, and it is heavily laden with food reserves, and therefore it has a lot of cytoplasm in a dense cytoplasm in it. And see the generative cell. The generative cell is a very tiny spindle shaped cell. In later stages, what happens is, let me draw the pollen here for you, only the inside of the pollen, not the outer structures. Outer structures, as you all are aware, there is intine. This is the inner cell wall. What I drew here was the plasma membrane. This is the intine or the inner cell wall. Okay, and then I told you the outer cell wall is very resistant and very protective. It is called as the exine, correct? Now in later stages, what happens is, this is the irregular nucleus of the vegetative cell. This generative cell becomes very flat, and it starts floating in the cytoplasm of the vegetative cell. See, what I'm showing you here in dots is the cytoplasm of the vegetative cell, and this spindle shaped or vermiform cell which is floating in the cytoplasm is what is referred to as the generative cell. So in the later stages, what happens is, the generative cell which was sitting on one side will get detached, and it will start floating in the cytoplasm of the vegetative cell. So this is the cytoplasm of the vegetative cell. Okay, and then you can see how the generative cell is freely floating inside the cytoplasm of the vegetative cell. So we now saw that there is a two-layered wall in the pollen grain. The outer wall is called exine, and the inner wall is called intine. I have labeled the exine and intine over here. The exine is made up of a very tough organic material, the most resistant organic material. A very important question in your board exam. It is called sporopollenin. The very tough organic material that is present in the exine of the pollen grain is referred to as the sporopollenin. It can withstand high temperature. It can also withstand strong acids and alkali. Till today, they have not discovered any enzyme which can digest this chemical called sporopollenin. That means it is such a strong chemical, and that's why it ensures that all the parts of the pollen grain within are kept safe. They are also kept free from destruction by high temperature and strong acids and strong alkali. Okay, so this is the structure of the pollen grain. So I hope it is clear to you. We saw, once again for your understanding, how many cells inside: a larger vegetative cell, a smaller generative cell. Initially the generative cell is sitting here at the bottom. Later it starts floating in the cytoplasm of the vegetative cell, as I've shown below here. And then what are the layers? Outer layer is the exine, inner cell wall layer is the intine, the innermost membrane is the plasma membrane of the vegetative cell. Vegetative cell stores a lot of food reserve. It has irregular nucleus. Generative cell is very small, fixed towards one side of the vegetative cell. Later it starts floating in the cytoplasm of the vegetative cell. So now we saw that the pollen grain is composed of two cells. That's why we call this pollen grain as the two-celled stage. That is, one vegetative cell and the second cell is the generative cell. And then I told you the inner cell wall was intine, which is continuous, made up of cellulose and pectin. And like I already mentioned, there are two cells inside a mature pollen grain, that is vegetative and generative cell. In 60% of all the angiosperms, the pollen grain is released from the anther. In what cell stage? In two-cell stage. In the remaining species of angiosperms, they are usually released in what cell stage? In the three-cell stage. What is this three-cell stage? In three-cell stage, what happens is, so this is the pollen grain. Okay, this is the vegetative cell nucleus. This is the cytoplasm of the vegetative cell, and I told you that inside the cytoplasm of the vegetative cell there is a generative cell. So this is a two-celled structure. The pollen grain is now at its two-celled structure. The larger cell is the vegetative cell. This is the vegetative cell, and the cell which is floating in the cytoplasm is the generative cell. Okay, so let me call it as, abbreviate it as VC and GC. So this is the vegetative cell, this is the generative cell. In some plants, what happens is this generative cell will undergo mitosis. I'm writing here. The generative cell will undergo mitosis. Now see how the structure of the pollen grain changes. The vegetative cell is as it is. It has an irregular nucleus, and this is the cytoplasm of the vegetative cell. Now I told you that the generative cell will undergo mitosis, so it will form two cells now which are a product of mitosis. So we have two additional cells now plus the vegetative. This is the vegetative cell with a nucleus and cytoplasm, and now we have two cells which are additional. So 2 plus 1 is, what stage is this now? This is called the three-celled stage. So in very few angiosperms, the pollen grain is shed off from the anther in what cell stage? In the three-cell stage. Otherwise, in 60% of the cases, it is shed off in what cell stage? In the two-cell stage. How did two-cell become three-cell? Because the generative cell, which was sitting in the cytoplasm of the vegetative cell, will now undergo mitosis to form these two cells. Do you know what these two cells are called? These are the two cells which are nothing but the male gametes or the sperm. They don't look like sperm because they don't have a tail. They are immotile. So these two cells which are formed by the mitosis of the generative cell are nothing but the male gametes. So in such a pollen grain, there will be a vegetative cell with an irregular nucleus as usual, and in the cytoplasm of the vegetative cell, there will be two cells which are formed by the mitosis of the generative cell. And what are these two cells now called? These two cells are nothing but the male gametes. And like I told you, this is the cell membrane of the vegetative cell, this is the inner cell wall called intine, and this is the outer ornamented cell wall which is called exine. There are parts where only intine is present and exine is absent. And what are these parts called? Like I said, these parts are referred to as the germ pores or the germ furrow. So in 60% cases, the pollen is liberated in two-cell stage, otherwise it is liberated in three-cell stage. Remember, the gametes were formed by the generative cell. And how did the generative cell form the gametes? By undergoing mitosis inside the cytoplasm of whom? Inside the cytoplasm of the vegetative cell. So a pollen grain is either two-celled or it is three-celled. So that's what I was telling you here. In 60% it is two-celled. In the remaining one, the generative cell undergoes mitotic division to form two male gametes before the pollen is released from the anther. If the pollen has two male gametes and a vegetative cell, we call the pollen grain now to be a three-celled stage. It is not a two-celled stage. Okay? So that is a very important thing. And another important thing is the pollen grain is also referred to as a male gametophyte. It is a name given to the haploid generation in the life of a plant. Remember, the pollen grain is haploid, and now we saw that in the pollen grain there is a generative cell that can produce male gamete. So it can be considered to be a highly reduced plant because it has only 3 cells in it, or when it is liberated it has only 2 cells, but it is a plant that is capable of producing male gamete. So we can call the pollen grain as what? As male gametophyte. Okay? Pollen grain are also known to cause lot of allergy and bronchial afflictions in patients, like in asthmatic patients, and people may grow severely allergic to pollen grain, say for example from plants like Parthenium. But in the Western countries, it has become a trend now to consume pollen in the form of tablets and syrups because it is said, they claim, to have enhanced the performance of athletes and race horses, because pollen grain is provided with rich food reserves which may be energy supplements to humans and animals, like you saw in race horses as well. Once the pollen is released, where should it go? Remember, anther. From the anther, we have the pollen that is coming out now. We saw that the pollen that is coming out is either two-celled or it is three-celled. Where should it go now? It should go and get deposited on the stigma of the carpel, isn't it? There is a female reproductive part with ovule inside, and this is the stigma. It should go and get deposited on the stigma. By the time it goes and gets deposited on the stigma, it should not lose its viability. Viability means its ability to fertilize the egg, its ability to germinate once it is deposited on the stigma. Suppose it is too late. By the time it goes and deposits on the stigma, then there is no use because the pollen would have lost its viability. Okay, so if it loses its viability, then it cannot fertilize the egg inside the ovule. It is of no use to the plant. What is the time duration of viability? Now, if you take plants like rice and wheat, the pollen will lose viability in very short period of time, that is 30 minutes. That means within 30 minutes it should go and fall on the stigma, otherwise it will not successfully carry out reproduction. Pollination will not be successful if it does not deposit on the stigma within, because beyond 30 minutes what will happen to the pollen grain? It will lose its function, it will lose its ability to fertilize the egg which is sitting inside the stigma, the egg which is sitting inside the carpel or the pistil, which we will discuss later. Ultimately, the pollen grain, as you all saw, is producing male gametes whose ultimate goal is to go in search of the female gametes sitting inside the ovule inside the ovary. So if they don't reach the stigma on time, they cannot fertilize. In some members of Rosaceae, Leguminosae and Solanaceae, we have pollen grain which can be viable for months. Even after months, if you collect the pollen grain, and even after several months, if you drop it onto the stigma, they can still carry out fertilization. So they are viable or functional, not just for a few minutes, like less than 30 minutes in cereal, but it is viable for months together. Pollen grain of large number of species nowadays, they are preserving the pollen grain in liquid nitrogen at minus 196 degrees centigrade, and these establishments where pollen grain is stored for future use in liquid nitrogen indefinitely, they are referred to as the pollen banks. They are used in the crop improvement program. You can transport the pollen grain from one country to another, and if the characters are favorable, it can be used to develop hybrid varieties in other parts of the world, and that production of hybrid varieties is a feature of improving the existing agricultural crops, so that we get a better yield and we also get a better quality of the yield, not just the quantity, better quality of the yield. Okay, so what did we learn in today's class? So in today's class, we learned the basic aspects of the flower and its parts, the essential parts, the non-essential parts. We learned about some of the events and structures which happen before fertilization. Specifically in the stamen, we saw in the stamen there is anther. In the anther, we saw there are compartments or cases, or there are chambers which are called thecae, and these chambers are called microsporangium. They have a tissue in the center called the sporogenous tissue, surrounded by several layers of walls. We saw the different wall layers in a young anther. Inside the microsporangium, we saw that the homogeneous cells, which are called the sporogenous cells, they themselves can behave as pollen mother cells. They can produce microspore through a process called microsporogenesis. What happens to the microspore? It develops into a two-celled or a three-celled pollen grain. In 60% cases, it is two-celled. In the remaining species, the pollen shed from the anther due to the rupturing or the dehiscence may contain in it three cells, that is vegetative cells and two male gametes. Homework for today: Now, two major essay type questions have been asked. One is describe the transverse section of the young anther, in which you have to show the microsporangium. You also have to show the enlarged view of what is there in the center, the sporogenous tissue, and the four layers which are surrounding the microsporangium in the anther. And then you have to talk about the process of define microsporogenesis. What is a microspore tetrad? Why is a typical angiosperm anther considered to be dithecous? Because I told you, you can divide it into two halves. In each half there are two compartments or two cases. In Greek, literally theke means case. Since in each lobe there are two cases, we can call it as a dithecous anther. And also a very important 5 marker, write a note on the structure of the pollen grain. For this you have to draw your own diagram. Like I showed you, you draw the diagram showing vegetative cell, generative cell, plasma membrane, the intine which is made up of cellulose and pectine, and the ornamental layer which is the exine made up of sporopollenin, and show the germ furrow or the germ pore where exine is not there but only intine is there. Okay, so with this we will complete this lecture, the first lecture in the chapter sexual reproduction in flowering plants. Thank you.